Department of Biomedical and Health Sciences at the University of Vermont. And he studies the molecular mechanisms regulating cellular immune activation.
Thank you so much for the invitation. If people can't hear me in the back, please be proactive about sort of flagging me, and I'll try to project, or I'll be more mindful to use the microphone. So I've been invited to begin just by sharing for a few minutes what my science journey was like. In high school, I was mostly an English and history guy. I mean, I took science, but it wasn't a passion of mine. And then I had a really incredible senior biology teacher who completely changed not only my interest in the discipline, but ignited a passion for teaching. It was a kind of teaching that I hadn't experienced before, and it got me so excited. And even though it wasn't my best subject, and that's how I knew that my feelings for it were real, because I was incredibly type A in high school, I fell in love with biology, and I became so interested really in the wonder of the cell. And that is sort of where I still am in my relationship to the field. I would say that my primary passion and interest is in this incredible wonder of how this basic unit of life, the eukaryotic cell, can take in relatively simple but many different inputs and make sense into it and translate it into what appears to be very complex behavior. And when you amalgamate all the different cellular responses into a complex multicellular organism, such as a mammal, you get this incredibly rich, responsive biological activity, which seems so much greater than the sum of its parts. And that's really what I became so interested in. And so fundamentally, I consider myself a cell biologist, and I just happen to do that in the field of immunology and study immune cells. So that's sort of the scope of my interest. So after high school, I didn't know for sure that I wanted to major in biology, but I wanted to keep the option open. And for many of you, you probably know that if you've chosen a specific field in science, you have to take all of these prereqs, some of which may be fairly painful for you. So for me, that was calculus and chemistry and physics, and those were all sort of the things I felt like I had to slog through to get the bio major. But I pursued it, and once I'd done all my prereqs, I was like, well, the bio is still so super fascinating. And so I ended up getting a double major in biology and philosophy to sort of satisfy my liberal arts tendencies and interests as well. The thing that really changed fundamentally my journey, and I will just say that I was a first generation college graduate. I did not go to college with the understanding that I was preparing for a job. I was really naive in that way. And this is so different from the modern culture, I think, of how many students enter college thinking about it, at least at some level of career preparation. So I really had that moment as a senior in my undergraduate career of, oh shoot, what am I gonna do after I graduate? And the thing that really changed for me in my college experience was I did undergraduate research, and I fell in love with it. And it started with a summer opportunity where I stayed on campus and worked in the laboratory of a professor on campus, and I found that I really was in love with the process of inquiry and the actual mechanics of doing research. And so as I was graduating, it seemed pretty obvious to me at that point in time that I would try to get a job as a research technician, because again, and this was a function of being a first generation college student, I didn't realize that for PhD programs in sciences, you get a stipend. I assumed you paid for it like you pay for undergraduate, and I had a significant amount of undergraduate student debt, and so it never even crossed my mind that I would go on to graduate school. So I looked for jobs as a research technician because I knew I loved to do research, and I started working in a lab, and the lab had graduate students, and about 6 months in, I overheard the graduate students talking about how they get a stipend, and it was one of those like record scratching, stop the music moments. I was like, what are you talking about? You're getting paid to get a PhD? And they were like, yes. And And I thought, why didn't somebody tell me this, sooner? So I worked as a research technician for 2 years, and then applied to graduate school and went to graduate school, and loved the research I was doing. And somewhat naively still, I imagined that after I finished my graduate work, I would immediately go and get a job at a small, primarily undergraduate institution like the one I got my undergrad degree at, and sail off into the sunset of the career of my dreams. And at that point, I had another moment where I didn't realize that there's this thing called a postdoc, which is a multi-year experience that's often expected for faculty positions, where you train almost like an apprentice after you've gotten your graduate degree for anywhere from 3 to 7 years nowadays under an established faculty scientist. And that's where you develop an independent research program, which you often bring with you into hybrid teaching research positions, which is what I wanted to do. So I went through all of that, and my research career was very interesting, but my primary passion was teaching, and I knew I wanted a career where teaching was a really significant component of that. Now, in normal graduate training, you don't get a lot of teaching experience. There's sort of a perfunctory TA-ship that you do for a couple of semesters. So while I was a postdoc, in order to really know that I wanted to teach, I ended up teaching several different intro biology laboratories at a local community college near where I was doing my postdoc, which was in upstate New York in Saranac Lake. And that's where I really confirmed for myself that teaching was something that I wanted to do, and so I started looking for positions where there was an equal emphasis on doing laboratory research and teaching. And so I got a job at the University of Vermont and started there 9.5 years ago, and I've been there since. And I really have the opportunity there to continue to sort of express both aspects of my professional interests in both the teaching side and the research side. Any questions about that? Yeah.
What colleges did you attend?
So the question was, what colleges did I attend? So I did my undergraduate at Amherst College in Western Massachusetts, and I did my graduate work at Dartmouth Medical School in Lebanon, New Hampshire. Is there another question? Same question? Yes. So my undergraduate was at Amherst College in Western Massachusetts, and my PhD I got at Dartmouth Medical School in Lebanon, New Hampshire. Sure. Okay. So today I'm going to tell you one of the stories, one of the research projects that we have in the laboratory. And there's various times that it'll get pretty granular and into the detail, and I really would welcome and invite any questions about anything that's unclear, because in this talk and in all of the teaching that I do, it's much more important to me that we talk about what we cover in depth and that we're on the same page than that we get through it all, because ultimately it doesn't matter to you or to me whether we hit every single one of my slides, but it's really just a story about science, and it's a story about scientific investigation, and really an opportunity to think about how we ask the types of questions that we ask and how we arrive at the types of answers that we arrive on. Because for me, the discipline of science, the details are great and they're interesting and they can be fun if you're into it. But the fundamental value as scholars, for all of us, is that we're learning a very specific type of modality about how we ask questions about the natural world, how we ask questions about the world in general, and how we ask them in a particularly rigorous and disciplined way so that we have a concrete understanding and a certain level of confidence in the answers that we arrive to those questions. Because science at its very heart is this really amazing type of fortune telling in a way, right? Because what we do with scientific information is we study correlations, we study cause and effect patterns in a particularly type of rigorous way, so that when we feel really confident, when we meet a certain bar of a cause and effect relationship, we start to be able to have this predictive power that when we see a predicted cause, we can predict the effect, right? And so this can happen with observation of the natural world, but it translates into all aspects of science, including those of medicine, right? What we're doing when someone comes in with a particular type of diagnosis or a set of clinical symptoms, and we give them a particular medicine, we're essentially looking into the future. We know, or we think we know, with a relatively high level of probability, that if somebody has some kind of condition and we give them the right medicine, that it will help them, right? So we're using this very rigorous study of the past under controlled conditions to be able to make meaningful predictions about the future based on the variables in play. And so that's really at the heart of what we're doing. We're just happening to be setting that discipline in the playground of one of the aspects of my research. So one of the central principles of immunology, and that's simply the study of the immune systems and the really complex set of barriers and cells and organs and the responses associated with these that allow organisms to protect themselves from outside threat or things that can damage their physiological responses, is one of the central aspects of immunology in general is this incredibly and tightly regulated balancing act between immune protection and self-preservation. And the one thing I want to understand, because it underlies the entire story that we're going to be exploring today, is that immune responses by their very nature are toxic, right? That is to say that our immune system, the cellular responses of our immune system, are designed to neutralize and eliminate pathogens or foreign invaders that can disrupt normal biology of the organism. Right? And these can be very specific, debilitating molecular mechanisms that kill bacteria, for example. But almost universally, there is some toxicity of those responses associated with the cells and the organism that makes those responses. And so the immune system has to be very finely regulated and threading the needle of how much toxicity is appropriate to neutralize the invading pathogen response without doing too much damage to the self. And so one of the things that you might appreciate is that when you get sick, right, if you have a cough or a respiratory virus and you feel awful, right, like your head is pounding and you're having a hard time breathing, most of those symptoms that you're experiencing are not directly caused by the virus itself. They're being caused by the intense immune response against the virus. Now, if left unchecked, the virus would replicate out of control and it would do its own damage. Right, but I'm illustrating this point because our immune responses have some self-toxicity in and of themselves. And this idea at the cellular level of how much toxicity is protective against foreign invasion versus too much and is self-damaging is at the heart of the story that we'll be exploring today. And the molecule that is at the center of the story is a antimicrobial gas called nitric oxide, which I'll introduce in more detail in a second. The cellular players at the heart of our story and are the central cells that I study and have studied for most of the last, oh man, it's 19 years now, okay. almost 2 decades here, are these beautiful cells called dendritic cells. And these are not to be confused with the dendrites associated with neurons in the central nervous system. These are dedicated immune cells which reside throughout the body. They're named, the dendra root means branches, and they're named for these branch-like projections which emanate from the cell body. And as happens so much in biology, form sort of follows function. And so what you might be able to predict from a cell that has all of this rich architecture and surface area on its outside is that these are fundamental immune sentinel cells. So their job, they reside in some low frequency circulating in the blood, and they also reside in your tissues. And their job is to constantly use these branch-like projections of their cell membrane to sample the local microenvironment in the blood and the tissues and recognize foreign invaders, right? And we'll talk a little bit about how that recognition occurs on the next slide. But basically their job is to ask the very simple binary question about their environment at any point in time, which is threat or no threat. And in the case of no threat, they exist in this sort of quiescent, low-level active state. They don't really engage in any of their immune function. But when they recognize threat, they undergo a very dramatic set of molecular changes and change their functional capacity in a very dramatic way. And they undergo this process of activation or maturation, which is at the heart of the studies that we do, trying to understand how that activation process is regulated. Any questions there? Okay. So I'm going to show you a number of studies where in controlled laboratory settings, we activate these immune cells by giving them the perception of threat, right? We basically mimic or simulate a bacterial encounter and induce these cells to undergo the processes of activation and maturation that are associated with these active states. So I've talked a little bit about the basic overview. For our studies, it's not super important for this class, but almost all of the immune cells in the mammalian immune system originate from stem cells in the bone marrow, and these cells are no exception, and we generate these on a weekly basis for our studies by harvesting the bone marrow from the long bones from the femurs in mouse legs. We flush out the bone marrow cells, which include the progenitor cells, and then we apply differentiation factors in a petri dish and an incubator and differentiate these cells over the course of a week to generate these dendritic cells for our studies. So how does this activation process occur? Dendritic cells and other immune cells possess many dozens of different types of what we call pattern recognition receptors. These are cell surface receptors. Some of these receptors are actually intracellular. And they have recognition motifs for what we call pathogen-associated molecular patterns. So there are certain molecular signatures that certain types of bacteria express on their cell surface that none of your cells express on their cell surface, right? And so at the heart of this intricate cell receptor system is the ability to discriminate one of the fundamental questions in immunology, which is self or non-self. And so when these cells recognize foreign molecular signatures, these non-self signatures which are enriched in bacteria and viruses, these cells become activated because downstream of these receptors are signaling transduction pathways which induce the transcription of specific protection-associated genes, which lead to the production of new proteins, which lead to new functional capabilities that these cells can now engage in, which directly correlate to their immune function. And so the two major stimuli that we'll be applying in the studies I'll show you today are either whole bacteria, which is simulated here in this cartoon, or a purified bacteria-associated molecule called lipopolysaccharide, shortened for LPS. And LPS is an enriched molecular signature in the bacterial cell walls of gram-negative bacteria. So when we give purified LPS to our cells, they respond as if they just saw a gram-negative bacterium, and they engage in the inflammatory pathway and the activation pathway as if they saw that bacterium. Any questions there? Okay. So what happens when these dendritic cells become activated? They undergo two major types of immune protective responses. The first one is they start secreting a whole bunch of soluble molecules that allow them to initiate this process of inflammation. It's basically like pulling the fire alarm in your tissues and saying the danger is here. Here is where. And so some of these secreted soluble factors recruit other immune cells to the site of bacterial encounter. Some of them are directly antimicrobial, like nitric oxide, which is going to be the central player of our story today. And some of them talk to other immune cells nearby and activate them, basically saying, hey, danger over here, and they're able to communicate to other cells molecularly and activate them, even though those cells nearby didn't see the danger directly. They experience it indirectly by virtue of these soluble chemical messengers. The other things that dendritic cells are very good at over here is that they're specialized immune cells that, let's say you get like a bacteria introduced into your skin from a puncture wound in your finger. the dendritic cells there will become activated, they'll pick up the foreign molecules associated with the bacteria, and then they will leave the site of infection and they will travel to specialized immune reactive centers called lymph nodes, where they engage in their second really important immunological function, which is their ability to interact and activate specific subsets of lymphocytes, T cells specifically, and those T cells have their own very important specialized immune function. So as such, dendritic cells play really important roles both at the site of infection and also to stimulate a really potent second wave of immunological response against foreign invasion. And we're really going to focus mostly on the role here, even though it informs what happens over here. So the lens with which we ask it, again, the fundamental questions that we're interested in is, how do these cells get activated? What regulates their activation? And can we understand the components of this activation so that we can understand how this balance between immunological protection and self-toxicity is maintained during these? And the lens with which we ask a lot of our questions is the lens of cellular metabolism. So when I was starting to develop the research questions that I would take with me when I started my job at UVM, so this was during my postdoc, the lab was very interested in understanding the nutrient requirements for this activation process. And a lot of the work that I'd done in the years that I was there was studying the importance of sugar metabolism, primarily glucose metabolism, as this really important fuel source, which was driving a lot of the questions. And we became very interested in some of the nuances, as in what is the time frame with which this metabolism change occurs. I'll talk more about this. What are the molecular signaling mechanisms that govern this? And really the fundamental question of, if we take away access to enhanced sugar metabolism, which we see when these cells become activated, what goes wrong? And when we find out what goes wrong, we can start making inferences and about pathways that are critically fueled by sugar metabolism that are important for successful immunological protection. So, this is the part where I try not to lose you and have your eyes glaze over as we go through some very, very rudimentary biochemistry. So don't worry at all about Krebs cycle and glycolysis pathway. That's not the point here. What I want to emphasize is that when we study biochemistry and we get our intro biochemistry or cell metabolism lectures, we'll often sort of trace the complete catabolism of a glucose molecule. And you'll often learn that the first step in consuming glucose and breaking it down is a process called glycolysis, which happens in the cytoplasm. And then under respiratory conditions, pyruvate gets converted to acetyl-CoA. It enters the mitochondrial TCA cycle reactions, and then complete oxidation of glucose-derived carbons occurs in the mitochondria, and we get a whole bunch of ATP coming out of that, and that's under ideal conditions when oxygen is available. And then we'll also often teach and think about the process of fermentation, where this process of glycolysis in the cytoplasm under anaerobic conditions when oxygen isn't available can occur, and you can still get some energy out of glucose that way. It's just not nearly as efficient as whether or not complete oxidation of glucose occurs. Does this feel at all familiar? Basic idea of this? Okay. So two central sort of metabolic fates of glucose. One is partial oxidation through glycolysis, and lactate is produced as an outcome of that, or complete oxidation through the TCA cycle, and we generate a lot of ATP. And the reason why I'm telling you this is because we have instrumentation that allows us to assess the balance of these two metabolic pathways and study how the emphasis or the usage of these pathways changes with activation. And the very short version of the story, which I'm not going to show you all the background on, but this is the premise for the work, is that activated immune cells enrich glycolytic catabolism of glucose, and you see that by an increase in lactic acid production and lactate accumulation in the media, where resting cells primarily engage in the full oxidation of glucose and primarily generate ATP through the mitochondria. And so one of the things we've become very interested for a long time is why do cells shift an emphasis to this pathway as opposed to this pathway, and what are the consequences when they can no longer do this, and why can they or do they no longer do this? And that's really a central component of the story that we're examining today. Any questions? Okay. So one of the things that we discovered relatively early on is that this glycolytic shift, which we talked about, this glycolytic reprogramming, this shift to glycolysis as the main utilization of glucose during activated conditions, it's really a function of two separate, there's two different causes for it. There's an early wave of glycolysis that happens immediately. And in this state, the cells are still respiring and they're doing perfectly fine. We know the phosphorylation events and the signaling pathways that mediate these changes. We know that this is during that early burst of activity where these cells are undergoing many different transcription and translational processes which are associated with their activation. And we also know that in addition to consuming extracellular glucose and importing it in after activation, they're also burning up at a higher rate intracellular sugar pools in the form of glycogen. And that was one of the things that was discovered by the first graduate student who worked with me at UVM, Pugh Thway, was the study of the importance of glycogen stores as an important fuel source for activation. And we know that if we inhibit this ability of these cells to rapidly increase their intake and use of glucose, we see a myriad of deficits in their immunological capabilities, mostly at the translational level. So these cells, if you inhibit their ability to use glucose after activating them, they can still turn on and express the genes and convert the DNA to RNA expression. But when it goes to converting that RNA into amino acids and protein production, the protein production gets severely impaired. And so all of those chemical mediators that I talked about that influence their ability to communicate to other cells, to recruit other cells to the site of inflammation, become severely impaired. And so there's dozens of different immunological deficits that occur if you prevent this early glycolysis from happening. Okay. And I just basically talked about this, right? Is that this early glycolytic reprogramming, it allows a specific type of metabolism occurs, which is associated with the protein production that is critical for normal immunological function. And when we prevent that from happening at a metabolic level, we see immune function deficits. So these things are intrinsically linked. And what I'd like to emphasize here for just a second is that this was very valuable. These kinds of studies, this field of what's now been dubbed immunometabolism, has been really valuable because it's allowed us another regulatory checkpoint by which we might be able to understand and influence immunological outcomes. And when we broaden out our understanding of immunology into the clinical realm, there are instances, scenarios where the immune system is overactive, and autoimmune disease is one aspect of that. And there are instances where the immune system is underactive and not doing its proper job, and cancer or uncontrolled infection is another example of that. And so from a really zoomed out perspective, we study these molecular and metabolic regulatory checkpoints with the hope of being able to understand the switches that push the immune system in one direction or another, with the ultimate hope of understanding it to a level that we might be able to harness this understanding in a clinically beneficial pathway. But that's zoomed way out, and now we're going back into the weeds of the molecular studies that we're doing. So I talked about how this glycolytic reprogramming is the function of two independent waves. So there's that early burst, which is associated with the pattern recognition receptor signaling that first happens when a dendritic cell encounters a bacteria or a viral associated ligand. And then there's this second sustained wave that occurs. It appears to be a permanent commitment to glycolysis and a permanent shutdown of mitochondrial respiration. And what we have come to discover is that it is exclusively and entirely dependent on whether or not these cells produce this antimicrobial gas called nitric oxide, which I'll introduce in just a second. Okay. So what is this molecule, nitric oxide? It's a toxic antimicrobial gas, which is made as a byproduct of arginine metabolism So arginine inputs into the chemical reaction, and citrulline is the metabolic byproduct, and nitric oxide is the other product that is made. Now, nitric oxide it's a lipophilic gas, so it freely diffuses in and out of cells and across all aspects of the cells, and it's highly unstable. It's a highly reactive compound, the half-life freely in the cell is on the order of seconds. By design, right? This is antimicrobial. It's supposed to be reactive because the way that it works is it's made by these cells which are encountering bacteria and viruses, and that gas then forms covalent modifications and molecular changes to the organic molecule architecture of pathogens and renders them non-functional as a result. And so that's great, right? The way that it works is these dendritic cells see gram-negative bacteria or lipopolysaccharide. They become activated. The signal transduction pathway downstream of these innate immune receptors turn on the gene which codes for the protein that makes nitric oxide, and that gene is called NOS2. The protein that codes for is iNOS, or inducible nitric oxide synthase. And so the gene gets turned on, the protein is made, and then this protein produces nitric oxide gas, which is then free to diffuse across the membrane, interact with pathogen, neutralize pathogen, and protect you from uncontrolled bacterial replication in this example. But there's another side to this, which is that nitric oxide is not only specifically reactive with bacteria and viruses, it reacts with all organic molecules. And so at some frequency, at some level, and this is central to the questions that we ask in my laboratory, the nitric oxide also feeds back on the mitochondria itself and inactivates and makes permanent and reversible changes to mitochondrial function. And it's this part right here, if I go back a slide, that's responsible for the sustained glycolysis. Nitric oxide producing dendritic cells make enough nitric oxide at certain levels of stimulation where they permanently inactivate their mitochondrial function in a nitric oxide dependent way, and so they literally can no longer breathe. And so they end up relying on glycolysis as the central pathway out of necessity. And so what we're really interested in is what is the optimal dose and how is it regulated that we can protect ourselves from bacteria in this host protective side of nitric oxide function and minimize the host cytotoxic effects over here. And we'll talk a little bit about some of these. Really, number one, the electron transport chain complex inhibition by nitric oxide, post-translational modifications to metabolic enzymes. We won't talk much about that. And we won't talk about the altered metabolic carbon flux either. Any questions? Yeah, wonderful question. So what's the question behind the question?
So hypothetically this happens and the nitric oxide goes back into the cell, cause the cell harm, would that cell eventually just kill itself anyway and be replaced? Or
Yes. So for those of you who couldn't hear, the question is, what's the big deal, right? Isn't this thing or this is too big of a deal, right? Why is the cell killing itself? Or is the cell going to kill itself? And what happens to the immune response if the cell kills itself? Does it not matter because it will just be replaced by other immune cells, or does it actually have functional consequences in a real life setting? And we'll get to that. The answer up front is that it is cytotoxic, these cells will die. And so what's happening here as it's regulated is the critical and important thing is how long these cells remain alive after activation. Because that second piece of their immunological function, the ability to go to lymph nodes and stimulate T cells, if the window of survival is, let's say, 24 hours after activation, they only do that function for a very short amount of time. So you reduce the probability that you'll activate the right T cells. But if you're not dying after one day, and let's say you survive for 3 or 4 days, you've increased the window. And so what we're really interested in is finding out what is the right level where the window is big enough so that they can do that critical function of their immune response. Yeah.
For your people that you viewed, did you just view I would say the average person? So for me, I know somebody with Crohn's disease, that's a immune it attacks your own immune system, so that would be a different response to what you're trying to
Yeah, you're asking like, is something like an autoimmune disease operating under the same types of activation principles? That's a really amazing question and very complicated question. So a lot of these autoimmune diseases are multifactorial, and they can be influenced by, let's just say in the case of Crohn's disease, the microbial environment in the gut. That's certainly a piece of it, in which case your immune cells are recognizing something that feels dangerous and starting to act there. But in the cases of a lot of these autoimmune diseases, something has been misprogrammed, and you have lymphocyte responses, you have T cell responses, which are often recognizing not just microbial microbes in the gut, but what we call autoantigen, a piece of normal self biology that incorrectly got triggered an immune response. And it becomes relatively hard to undo those when the T cell response gets misreprogrammed in that specific way. But the reality is that a lot of autoimmune diseases actually occur when we have chronic, long-lived, low levels of inflammation to something. And that can be tissue damage, it can be microbe-associated, so it is absolutely related. It's just not a one-to-one comparison with what we're talking about here. But great question. Yeah, so our key interest is in the balance here. So this is the basic model that our experiments query, right? We stimulate our cells, the NOS2 gene gets upregulated, iNOS protein gets expressed. It's not expressed in unstimulated dendritic cells. Nitric oxide is then made when iNOS protein is expressed, and now we can query its antimicrobial function and its host cytotoxic function, and we can ask. And this is sort of a cool thing that Julia Snyder, who did most of the work I'm showing you, she got her PhD and worked in my laboratory last spring. But mitochondria, as far as we understand it, are essentially ancient bacteria that got incorporated, and it's sort of like the evolutionary origin of the eukaryotic cell was this sort of fusion event of two cells. And so mitochondria are bacterial in nature, and so in some ways it's not surprising that this innate immune mechanism, nitric oxide, which evolved for antibacterial, also feeds back and impacts our mitochondria, which is a cool conceptual idea.
So the nitric oxide only kills the mitochondria, like not like the cell wall of the dendritic cell or the other cells?
Yeah, so it's a beautiful question. The question is, are the mitochondria the only thing affected? And the answer is absolutely not. They're the most dramatic thing that's affected. And one of the things that we're actually studying right now, and I think it will become evident from the data that I get to, if I get to it, is that there seems to be this cascade that's very much dependent on the time and the dose of stimulus and how much nitric oxide is being made at any point in time. And certain things are hit more at lower doses than others, and so we're just beginning to actually map that. We become very interested at moderate doses of stimulation and moderate doses of nitric oxide. What are the first targets that get impacted? What are the secondary aspects that get impacted? And we know enough right now that some metabolic enzymes are targeted before the mitochondria are affected at all. But then the mitochondria are impacted by like a second wave, the next level of nitric oxide, and then so on and so forth. But we do know that the cytotoxic function, the cell survival piece, is entirely dependent on the respiration piece. So they die because the mitochondria can't work, and only because of that, it seems. Okay. And I don't want to go into the details here, except nitric oxide has many targets, and this goes a little bit to the last question, but here we're just in the world of the mitochondria. So 3 of the 5 enzymatic complexes in the mitochondria that drive the electron transport chain are known to have irreversible post-translational modifications driven by nitric oxide. And the one we're going to focus on is this reversible one, where nitric oxide actually competes with complex 4, cytochrome C oxidase of the electron transport chain. And it basically functionally competes with oxygen in this oxidase enzyme and prevents it from doing its normal function. And we know it's reversible because if in real time we put in a nitric oxide synthase inhibitor, all of a sudden the cells start breathing again. Okay, so this gets a little bit at the cell survival piece. So if we look 4 days after stimulation, here we have cells that are unstimulated. Almost all of them are alive. If we look at normally stimulated cells, all of them are dead by day 4. But if we stimulate cells in the presence of an INOS inhibitor, so the cells become activated, they do their thing, they just don't make nitric oxide, cell survival is completely rescued. So we know that there's a one-to-one correlation. If the cells make nitric oxide at a certain level, they're all going to die. And if you prevent that from happening, you're going to save them. And so now we have this interesting question that I alluded to a few minutes ago, which is how long they survive is directly proportional to the magnitude of the immune response that they're able to generate. And so we become really interested in the nuances of that regulation and how that impacts the immune response at large. Okay, and this is just another model of the same central question, right? Nitric oxide production. It can be antimicrobial and protective, but it can also lead to loss of mitochondrial respiration, which ultimately leads to cell death, less time to stimulate the immune response. What are the nuances of this balance? So now I'm going to talk a little bit about one of the genetic tools that we use. There are 2 different mouse strains to study this from a zoomed out perspective, because all of the dogma I just told you about the model and the system that we use has been studied in a single mouse strain, right? So we know, like anybody who's done research in model organisms or in mice, for example, and then tried to apply those same findings to the diversity of human samples, often becomes incredibly frustrated because it's just like, why are humans so different from one another? And it's because we're so genetically different from one another. And so there are huge and important and meaningful nuances in the way each of our immune systems would interact to the same given stimulus. It's the same reason why vaccines don't have the same efficacy in everybody, and there's like a broad range. And why a household that all gets the same viral infection, some people might be laid out for days and other people might just have the sniffles but be okay, right? There's this huge heterogeneity that's largely driven by our genetic differences, which underscore and are functionally important for the immune responses that we're able to generate. Now, in model organism and mouse research, for example, we will study a single mouse strain, and the way these are generated is that they come from an inbred population of mice, back-crossed, basically brother and sister mating back crosses for at least 20 generations. For many of these, we're talking of hundreds of generations of mice, because the founder mouse strains have been around for over 150 years, or over 100 years at least. And these populations are genetically identical. They're basically like identical twins. Every single mouse that's studied from a single strain has the exact same genetics as every other mouse that comes from it. So essentially what happens is we can study a whole bunch of mice, but from a real genetic diversity standpoint, we're just studying many clones of the same organism, right? The same genetic background. And so all of the studies that I just told you and all of the findings that I just shared with you were studied in a very common, widely used inbred mouse lab strain called the C57 black 6 mouse, right? And classic lab strains are grouped like this, and it turns out that these are all highly, highly related to one another, and so they tend to yield the same experimental results as one another when you go from one common laboratory strain to another one. But we wanted to ask a different question, which is, these nitric oxide responses that I've just shared with you that seem to be hardwired for what happens in the mouse, is it just true of the mouse? Or is it just true of the simplified genetic background that we happen to be studying? So there are these wild-derived strains, which are also back-crossed and genetically homogeneous. They're just brought out of, they've been captured from the wild more recently, and so they're genetically diverse and distinct from the common laboratory strains, right? And so we picked this one, PWK-PHJ, which you'll see called PWD in these studies. So they're also fully inbred and genetically identical to themselves, but they're genetically distinct from the common laboratory strains. And that's illustrated here. So this represents the black 6, and the brown mouse represents the PWD mouse. And what you can see if you do a genetic analysis called polymorphism analysis, or single nucleotide polymorphic percentages, you can see that the common laboratory strains, which are here in yellow, are very similar to each other with 15% or less differences between one another. But when you compare the PWD to the black 6, you're more than 50% distinct, which means that at a nucleotide level, at the DNA genetic level, the PWD mouse is 50% different than the Black 6 mouse. And so that's much more representative of the kind of genetic differences between each of you and me, for example, right? Or each of you from each other. And so by comparing these two very genetically different organisms, it allowed us, we predicted it would allow us to get some insight onto whether these nitric oxide-driven immune responses were common across the board or were specific to the genetic backgrounds that had been studied up to that point in time. And so we asked the simple question, right? If we stimulate the common laboratory strain versus the wild-derived laboratory strain, do they have the same nitric oxide response? And to do this, we can measure, remember I said the nitric oxide itself is a very unstable gas, but one of the reactants that it readily forms to is nitrite, and that accumulates in the media and is readily detectable and is indicative of the nitric oxide and proportional to the amount of nitric oxide gas that was made. So now we're comparing the common laboratory strain Black 6, B6, and the wild-derived laboratory strain, the PWD. Unstimulated, nobody makes nitric oxide. That's the control. We knew that that would happen. And then we asked what happens when we stimulate them. And lo and behold, the Black 6 makes its normal, robust level of nitric oxide as indicated by a high level of nitrite accumulation in the media. But the PWD animal makes way, way less. It's not zero. It makes some. It turns on the gene and generates some, but it makes much, much less. And so we became really interested in trying to understand that, right? And now when we look at the mitochondrial respiration, here's the core data. And before I get into this, I just want to describe that we have this instrumentation called an extracellular flux analyzer, which allows us to measure in real time oxygen consumption rates and respiration rates in these organisms. And the details of this assay are not super important, but what happens is we're measuring oxygen consumption rates. That's what OCR stands for, or OCAR. And this assay, which is called the mitochondrial stress test, we basically inject a series of 3 different mitochondrial toxins, which perturbate and disrupt mitochondrial respiration in various ways. And the take-home message is that when a cell fluctuates after each of these injection points indicated by these vertical dotted lines, it says that the cell is, the mitochondria in that cell are respiring. And when the respiratory curve is flat, it basically means that there's no mitochondrial respiration in the cell. So if we compare Black 6 to wild-derived animals, we can see that in the unstimulated state, dendritic cells from both strains respire perfectly normally, even though they are different from one another, and that's interesting. But in the LPS stimulated state, the wild-derived are perfectly respiratory, but the Black 6 have lost all of their respiration. And that directly correlates and is driven by the differences in the nitric oxide production between these 2 strains. Everyone with me there? Any questions? Yeah. Is that a question?
So, are you doing this test? You're using the cell from the femur up on this little under?
Yes.
So you don't really know, if the mouse is dealing better with the illness, symptomatic.
That's a great question. We're working our way towards those kinds of studies, right? So we start with the cellular response to like purified bacterial ligands. Then we move towards live immune cell, whole bacteria, live bacteria interactions. And then we move towards actual infections.
Now that's
Yeah.
That's a huge.
Yes, and we'll go into some of the complications of the system and some of the nuances to try to get more confident at our predictive power of actually going into live animal studies to a point where we might be confident to predict. We think that the wild-derived animals will do worse, let's just say. Okay, great question. So there's a problem here, right? And it's a problem and a strength, right? We intentionally chose an organism to study that's really genetically different. But it means that there are thousands and thousands of genes that are very different between these 2. And any number of those genes could be responsible. And we wanted to try to make a more definitive claim about its differences in nitric oxide that are driving this difference. And so we used a genetic tool called a consomic animal, I'll explain that in a second, which allowed us to genetically map or get closer to genetically mapping which genetics are responsible for driving the differences that we're seeing. So consomic animals are essentially animals where through a very complicated set of screening and back crossing the two different strains, and there's a whole library of these that are made, is that they've generated black 6 animals that either have only one chromosome of the PWD background in them or pieces of a chromosome. And this one right here, called 11.2, has a segment of chromosome 11 that contains the NOS2 gene, that contains our gene of interest. There are other genes in there as well, but it's a much smaller genetic interval than the whole background, on an otherwise black 6 background. So these 11.2 animals are genetically black 6 animals, except for this region of chromosome 11, which also contains the gene that codes for inducible nitric oxide synthase. And so what it allows us to do, from an experimental standpoint, is eliminate the confounding effects of all of that genetic heterogeneity between the two strains and shrink the locus of investigation down to this piece of chromosome 11, trying to get to the point where we can meaningfully infer that the differences are being driven by the differences in the NOS2 gene itself and not by some other genetic factors. So, what happened with the 11.2? Well, it's actually starkly different from this. It's not all or nothing, or almost all or nothing as we see here. It's something and intermediate, and it's also dose dependent. And so I'm going to take you through these data a little bit. So here again, we're measuring nitrite as an indication of how much nitric oxide was made, and we're looking at a dose of different lipopolysaccharide stimulation concentrations. And so what we can see, I'll just take you over here, is at high concentrations of stimulation, the black 6 and the 11.2 consomic animal are exactly the same. They make the same amount of nitric oxide at really high concentrations. But as you titer down the stimulus dose, right, and you get into a more nuanced portion of stimulated cells that aren't maxed out, you start to see these differences tease apart. And even at very small differences in nitrite accumulation here, when we look at the correlating mitochondrial respiration here, you can see that the 11.2 cells, which make a little bit nitric oxide, a little bit less nitric oxide, are still respiring. But the black 6, which are making just a little bit more, are now not respiring at all. And so what this system has allowed us to uncover is that there's this really finite and discrete threshold. There's a nitric oxide threshold where after a certain level being made, it stops all mitochondrial respiration, and below that threshold, the cells are still able to respire. And we can make a bunch of predictions based on that phenotype, which we'll get into in a second. But we've become incredibly interested in this respiratory threshold and trying to understand, because now this balance question really matters. Right? Now we've gotten to the interesting part, because at this level, at this regulatory level, maybe they're making enough nitric oxide to be antibacterial, but not cytotoxic. And at this level, maybe there's no extra benefit into the antimicrobial function, but now you're cytotoxic. And so we could imagine there would be this evolutionary pressure with regards to nitric oxide production regulation to sort of be at this level, where the cells are still respiring and still making enough nitric oxide to protect against bacteria, but not so much that they're shutting down their own cellular survival. Okay. It's 5 left?
5 left. We go to 5:50. That's
Sadness. Okay. So this is the data you just saw, and this data is just showing that there, besides nitrite accumulation, there's a more nuanced way to do this, which is to measure the biochemical product of the nitric oxide reaction, and that's citrulline outcome, and we can measure this by mass spectrometry, and it's just showing you that we can measure that even at low concentrations, excuse me, of LPS stimulation, we're getting nitric oxide being made, even if we can't measure a lot of it using this assay. Okay, so here we're illustrating the mitochondrial respiration more clearly at the different doses of stimulation. Unstimulated, both black 6 and the consomic animals both respire. At the high concentration, neither of them do. And at the intermediate concentration, the 11.2 cells maintain their respiration, but the black 6 are gone. So this really does matter for the cell respiration. And we would predict that this correlates to cell survival, and of course it does. So unstimulated cells, almost all of them survive. At low doses, only the 11.2s survive, but both populations are rescued by an INOS inhibitor. And at high concentrations, both strains die, and both are rescued or partially rescued by an INOS inhibitor. So I focused a lot of my talk on the cytotoxic side, but what about the immunoprotective side? So here's where we went to the next level, and we added live dendritic cell, live bacteria interactions, and asked the ability of the dendritic cells to control bacteria. And the bacterial infection we used was Listeria monocytogenes. This is the pathogen that you'll see like a couple times a year. You'll see a headline like lettuce recalled because of Listeria outbreak. This is just a foodborne pathogen that is associated with soil. And we use it because this is famously known and understood to be regulated by nitric oxide. Like when you have nitric oxide production deficiencies, organisms have a harder time controlling this bacterial infection. So in this situation, we used 2 different strains of Listeria, even though we couldn't measure significant differences in nitric oxide. And this is because of the lack of sensitivity of this particular output to measure very nuanced changes, we believe. We can see that whole bacteria, in addition to that purified ligand, still exhibit differences in mitochondrial shutdown. The black 6 respiration is way more suppressed than the 11.2, and this really confirming the presence of our experiment. In situations where the cells have molecularly favored respiration, they've become worse at controlling bacterial replication, because the respiring cells, which make less nitric oxide, have a much harder time controlling bacterial numbers. And so this is colony-forming units of bacteria, and you can see that the black 6 have much fewer bacteria than the 11.2s, and that some of this difference is diminished when you do the same experiment with INOS inhibited. And the same is basically true for both strains. Okay, so I just want to finish one thing, which is looking a little bit towards the future. So one of the things we become really interested in is when we look at the genome differences, the genetic differences between the black 6 and the wild-derived NOS2 allele, there are 6 non-synonymous amino acid changes between those 2 genes. That means there are 6 amino acids that are different in the wild-derived INOS protein and the black 6 INOS protein. So one of the graduate students in the lab is actually now working through and creating, basically artificially expressing this protein, both this version, and this version which with each of these substitutions changed one at a time, and being able to ask, is one of these amino acid changes the one that's responsible for the differential activity of the protein differences between these 2 strains? Okay. So just to summarize, we've established that there's a nitric oxide mediated threshold for respiration that controls both cellular metabolism and cell survival and bacterial protection. We know that there are multiple levels of regulation of both the gene and the protein through our different genetic systems. And we know that the previously established literature using just one mouse strain is not representative of the whole series. It just gave us one snapshot of, let's say, one organism. And it would be like studying a single one of us in this room and saying, that's how the human immune system works for everybody. And so the situation is obviously more complex, and so we've become interested in exploring the heterogeneity across different genetic landscapes. So the future directions, we're interested in examining some of these differences and really understanding more detail about the immunological impact and consequences of these nuances in regulation. So before taking questions, I just want to acknowledge the great people who've done this work. So Soyan and Bei are the two active graduate students who work with me in the lab right now. Soyan is the one who's carrying on this project, which Julia did. Here are our lab alums that have worked on various projects. I want to specifically mention Julia Snyder because most of this presentation was actually the slides she put together for her dissertation defense, and a lot of the work is the work that she did over the 5 years that she was in the PhD program at UVM. Our work is heavily supported by the Vermont Center for Immunobiology and Infectious Disease, with lots of funding and support from the leadership there. The graduate program I'm associated with, which is directed by Matt Pointer. This is Julia's thesis committee, and the funding sources which make scientific research possible. And I'm very happy to take questions at this point. Yeah.
What was like the initial question? Was like your study question that you were supposed to answer, or you were trying to find the answer to?
What was the initial study question? There were a couple, but I think the one that you're trying to get at is the fundamental question from the primary data that I showed you today was whether genetic differences between organisms, and by that I just mean different strains of mice in our context, impacted and correlated with differences in nitric oxide regulation to immune cell activation and functional cell and molecular consequences downstream of that. So really, do genetic differences or does genetic variability impact the way NO production is regulated, and what does that mean for metabolic and immune responses? Yeah. Mm-hmm.
I heard the term T-cell mentioned. What does that mean?
Okay, great question. So the question is, what do I mean by T cells? So if we think about the architecture of the immune system as a whole, just like the complex network of protective responses that our bodies generate, we functionally divide it into two major branches, what we call the innate immune system and the adaptive immune system. The difference between them is the innate immune system is a series of barrier and cellular defenses which serve as gatekeepers for that self-non-self response. And these innate immune systems are very quick, they're hardwired, and they don't learn. They don't have what we call immunological memory. They basically trigger the same way every time. So if you get infected by a bacteria today, and you get infected by the same bacterium a year from now, your innate immune response will trigger the exact same way. I'm oversimplifying a little bit. The adaptive immune system has that exquisite specificity and the beauty of the mammalian, really the vertebrate immune response, which is memory. And memory is driven by two major classes of what we call lymphocytes. T lymphocytes, which are a specific subcategory of these, and B lymphocytes. So when you hear about B cells or antibodies, B cells primarily exert their immune protective function by making antibodies. T cells exert their immune protection from cell-to-cell interactions. But T cells cannot get activated by themselves. They actually need help from dendritic cells to become activated. And so if you only had an innate immune response, you would lose all of the benefits of the memory response, and vaccines would be completely useless, right? Because the principle of vaccines is to essentially artificially educate your memory response, your T and your B cells, so that you have a standing repertoire of pathogen-specific fighters on hand for the rest of your life or for some window of time. So dendritic cells are this critical bridge between the innate and adaptive immune system because they are innate immune cells, but they're also the key to activating important types of T cells. And so without dendritic cells, you get both impaired innate immune responses and impaired T cell responses. And so I mentioned them here. If I'd given sort of the full talk, I mentioned them here because when we study dendritic cell impairment, it often will correlate to T cell activation impairments. So getting back to a previous question is, does this matter? Well, we know it matters because if we, for example, inject activated dendritic cells from different states into live animals and say, go ahead and do your thing, stimulate T cells, the T cell response will be dramatically be impaired in certain metabolic conditions of dendritic cells compared to ones that saw normal dendritic cells. Remind me when I said that.
So the process of introducing back into the cell.
Oh.
Do you think that has anything possibly to
Yeah, so this is a really, really great question. So the question is whether or not the cytotoxicity could possibly be linked to cancer. And the short answer, there's a lot of answers to that. One, humans make much more restrained nitric oxide responses than mice, than the black 6 mice. And in fact, one of the things that got us so excited about this, because people haven't really fully understood why, is that the genetic sequence of the wild-derived mice looks much more like human than like the other mouse strain. And so we believe that a more restrained nitric oxide production is ultimately the more beneficial way to do it, and to survive as an organism on this planet, and we're interested in following up that link. But the broader question of whether these kinds of self-cytotoxic responses can lead to cancer, the short answer is absolutely, because cancer is just a genetic disorder that is a sort of group of genetic disorders, whereby normal genetic and molecular machinery gets mutated and leads to a functional loss of control of cellular replication. So any cellular and molecular process that leads to mutations or altered function of core cellular processes has the probabilistic potential, just randomly, to hit the right target and induce a series of changes that can ultimately lead to what we call cellular transformation, or the transition of a cell becoming normally controlled in its cell cycle to becoming out of control. And I would say this is not unique to nitric oxide. Inflammation in general is strongly correlated with oncogenic onset. So any situation, any chronic inflammatory disease or inflammatory condition increases the probability of cancer emergence because of the inherent toxicity of inflammation in general, right? When we get sick, right, we feel bad because our system basically triggers this potent firepower and says destroy, right? Hopefully in a controlled way. But we know that this is not controlled when we think about, like early in the pandemic, for example, when patients were dying from toxic cytokine production. What was happening is that the immune response was so out of control, and these high levels of inflammatory molecules were in the bloodstream, that the body was overwhelmed by its own immunological response, which can lead to multi-organ system failure and death, right? So inflammation always comes with some price, and usually it's worth the price. But if it's very extravagant and acute, it's a problem. And if it's low level but unresolved and goes for a very long time, it's also a problem. When I was in graduate school, one of the things we were taught was that cancer is basically an unhealed wound. And it's a little bit poetic, it's nice poetry, but there's some truth to that, because many cancer originates in states where normal clearance doesn't happen. And the underlying understanding of this is that our immune system is very good at clearing abnormal self cells. There's whole branches of the immune system which are dedicated to that. And we first started appreciating this during the early wave of the HIV epidemic in the 1960s and 70s, because HIV positive individuals were all of a sudden showing up with high frequencies of cancers, right? And the inference from that, which has borne out to be true, is that our immune system is constantly monitoring and eliminating precancerous cells. And when you lose immunological oversight and surveillance of that, cancer pops up more frequently. Any other questions? Mm-hmm. So the question is there a therapeutic use for nitric oxide? And I would say maybe once upon a time, in the sense that let's actually talk about cancer, because it's one of those situations where we want, in cancer therapy, we want to be cytotoxic. And honestly, in the world of cancer therapy, we have historically played the same game that the immune system plays, right? We all know just from anecdotal or knowing someone with cancer that many cancer therapies are highly, highly toxic, right? People lose their hair, people feel sick all the time, right? People experience dramatic symptoms of toxicity. Because with first generation cancer therapeutics, we were essentially doing the same thing nitric oxide does, which is how much of the cancer can we kill without killing the patient, right? That's really the game that we were playing. And as we've gone to second, third, and fourth generation cancer therapies, the whole movement in clinical medicine in general is sort of specialized targeted medicine and trying to reduce the toxicity and increase the therapeutic specificity for the approaches. So maybe when we were playing more fast and loose with our patients, because we simply didn't have a better alternative, you could imagine a scenario. There are biochemical nitric oxide donors, which you can inject into animals or introduce to cells and just spew lots of nitric oxide with a relatively short window of activation. But I don't see it having a real place in the clinic now because there are so many like incredibly amazing and delightful specific mechanisms that are constantly being invented. Like the world of immunotherapy is just a wonder. We could easily spend an entire year talking about it.